Greetings everybody, this is Scott Tubbs. I'm going to be talking to you today about rotating soybean with peanut and that effect on pod yield, grade, and root galling from peanut root knot nematode. I'd like to acknowledge my co-author Patty Temper from USDA ARS in Tifton, Georgia. First of all, let's start off by talking about standard rotations on peanut. When we're talking about a standard rotation, then we're typically talking about peanut being planted within every 4 years since the last planting. And most of our farmers in Georgia are using either a 3-year rotation or a 4-year rotation, which would a 3-year rotation would refer to 2 additional summer crops of different crop species until you go back into peanut. The questions that we often get from growers are how many years should they leave between peanut crops? They also ask, do the crops in rotation with peanut matter, especially with other legumes? Some question which pest species are affected, and are some affected more than others by rotation? And then what management tools are there available aside from rotation to help manage certain pests and improve production? So the objectives of this study were to determine the effect of crop rotation sequences on peanut yield, grade, and root galling by peanut root knot nematode. I'll refer to peanut root knot nematode as PRKN throughout the remainder of this talk. And we're going to determine that effect on crop rotation via the different crop species that are grown between peanut and the duration of the sequences for each of these rotations. In addition, we had a second objective to evaluate peanut root knot nematode susceptible versus peanut root knot nematode resistant peanut cultivars that are currently available as runners in Georgia. The year of this study was 2018. Of course, this is the completion of the cycles was in 2018. The study had been running for a number of years prior to that in order to run through the full sequence to get to the completion year where all rotations cycled onto the same peanut year in 2018. The main effect was our rotation sequences. We had 9 different treatment sequences that we were testing. This included crop species, which of course included peanut. We had continuous peanut as our check. We had 7 of the crop rotations included corn in the rotation as kind of our standard rotation partner in comparison to peanut. We had some crop sequences that included soybean as our other legume crop. Soybean is grown in Georgia. It's not a high acreage crop for Georgia, but it is one of our 4 primary row crops, agronomic row crops that are grown in the state. Cotton being the other one. Did not have cotton in this trial. We used corn as our standard rather than cotton in this study. And then we did have a weedy fallow check as well, where the 2 years prior to the peanut cycle year, there was no crop grown, just the land was left fallow and weeds were growing in there. Then we had duration sequences for these various options. Now, naturally, all of these sequences were not duplicated in a factorial for all possible sequence combinations. That would have been too many to keep up with based on land availability. But we did have either 0, 1, 2, or 3 different summer crops between peanut plantings, and of course 0 being a continuous peanut rotation. And then in the terms of corn, we did have 1 year, 2 year, and 3 years of consecutive corn before going back to peanut. Soybean was in certain combinations, but we certainly did not use soybean as the sole crop in the rotation for a 2 or 3 year between peanut planting. That is not a common rotation sequence for our farmers, so it was not evaluated. Our sub-effect in this trial, it being a split plot, was peanut cultivar, and we used Georgia 06G being the standard peanut cultivar for Georgia. but this is a peanut root knot nematode susceptible variety. The picture on the right of the screen is a root that was pulled from a Georgia 6G peanut in a continuous peanut rotation where we had tremendous root galling occur from peanut root knot nematode. And then we had Georgia 14N as a peanut root knot nematode resistant cultivar in this study. The picture at the bottom of the screen is not of the Georgia 14N. This is another picture that I used, but it does show peanut nodulation of standard roots that are not root galled. This is just a heavily nodulated root. Most roots showed some combination in between where there was not quite as much nodulation, but there wasn't root galling to this level. So let's look at some of the results from this trial. First of all, we're going to talk about pod yield. And when we're looking at the rotation sequences here, all of the figures that you're going to see in our results slides will be set up the same way with the crop rotation sequences across the bottom on the y-axis. And we designate corn as a C, the fallow are the F, P is peanut, S is soybean. And on the left of the screen, we have our weedy fallow plots right here. And then, of course, our corn-only rotations where we had our 3 years of corn before we went back to peanut, 2 years of corn going into peanut, an alternating corn-peanut rotation. And then we have our sequences where we included soybean. So we had alternating corn, soybean, corn, peanut, so that there was a non-leguminous crop in between each of the legume crops. Then we had 3-year rotations between peanut where we had 1 year of soybean and then 1 year of corn with corn preceding peanut. Then we had the opposite rotation for that 3-year where the corn and the soybean flip-flopped and soybean preceded peanut with corn following peanut. Then we had our alternating rotation, soybean, peanut, would not be a consistent rotation in Georgia. Most farmers are going to include a non-leguminous crop in their rotation. And then, of course, our continuous peanut rotation is out here on the far right. Looking at these yields, there were statistical differences at the 5% level where we noticed that our soybean peanut rotation was our lowest yielding by far statistically of all the rotations in this trial. And our peanut where we immediately followed soybean also was statistically lower than our best yielding rotation, which would have been the 4-year rotation of corn, 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 and peanut. So this holds true to what we would recommend in Georgia, that our longer rotations are your best bets. But in this study itself, when looking only at the standard rotations without the additional legume, taking soybean out of the equation, there was no difference in this rotation trial for the length of rotation. So it was really just crop species that came into play for this rotation on this trial. Now taking a look at our grade data, total sound mature kernels. Once again, our soybean peanut rotation had our lowest grade. This was the only statistical difference at the 5% level for this trial. We were very close at the 5% level to getting additional differences, but we did not quite reach it at the 5% level. Had we run at the 10% level, then we would have also picked up differences using the continuous peanut rotation and the 2-year rotation corn peanut alternating compared to our top 3 grading treatments for total sound mature kernels. Now looking at the root galling itself, there was actually an interaction between rotation and the peanut cultivars, the Georgia 6G being the susceptible cultivar. So the data being shown is only the Georgia 06G peanut data. The Georgia 14N had 0 galling. So first we're going to take a look at the Georgia 06G peanut and the galling that occurred in this slide. So we saw our largest galling occur in our weedy fallow plots where there were numerous species of plants that could have been hosts for peanut root knot nematode in that field over the years. Of course, our soybean peanut rotation and our continuous peanut rotation also had very high root galling numbers, statistically greater than our other rotations. But all these other rotations that included corn in the rotation somewhere in the process did reduce our galling in comparison to those other treatments. I also want to bring in to the effect our peanut root knot nematode incidence compared to Georgia 14N. Anything with a star means that that rotation had higher galling incidence with the Georgia 6G peanut compared to our essentially zero incidence with Georgia 14N. So our rotations with the lowest galling numbers, which would be our standard corn peanut rotations, did not have any difference using Georgia 6G peanut compared to 14N. But as we got into poorer rotations, we did start to see a difference between having that susceptible variety in the rotation compared to a resistant variety. So those with the stars next to them would warrant some sort of additional means of reducing peanut root knot nematode incidence for Georgia 6G peanut if Georgia 14N was not available or not selected to be used. So in conclusion, going back to our original questions, how many years between peanut crops? Based on this data, we would say it depends on the species. For this trial, we saw no difference on our standard rotation crop using corn as our check. But if we were looking at a soybean rotation with peanut, then we were certainly affected. If you go back to literature, we would say that at least 2 additional years in most rotation studies, or the equivalent of putting peanut in the field every 3rd year, is our minimum, and the longer that you can advance that rotation, the better. Do the crops in rotation with peanut matter, especially other legumes? This data did support that, yes, that does matter. Soybean is a poor rotation partner with peanut based on the data that we had here. And that confers with other rotation studies that have been published. Corn and cotton are our most adapted species for rotating with peanut in our peanut producing areas in Georgia and also throughout the southeast. Are some pests affected more than others? Yes, certainly they are. Some pests are greatly influenced by rotation while other pests are not. Of course, we used peanut root knot nematode as our evaluation point in this study, and it was strongly influenced by the rotation. Some crops are hosts, others are not. Corn in this case was not a host, and soybean was a host for peanut root knot nematode. And what management tools are there aside from rotation? Well, we tested cultivar selection, and that did affect peanut root knot nematode galling in this study. But in terms of other pests, certainly pesticides, planting date, tillage, plant population, there's numerous factors that can affect various pests and overall production, depending on what pest we're talking about and how you can control it. That concludes the information in this study. One thing I did not point out earlier was that there was no yield difference between our varieties in this study. We did see the difference in the root knot nematode galling, but it did not translate to a yield difference. But all in all, I certainly want to acknowledge the support that we had from Dr. Tubbs and Dr. Temper's program teams over the years for data collection. We acknowledge our support from National Peanut Board and the financial considerations that were supported in creating such a long-term study over the years, and naturally the support of the Georgia Peanut Commission. Seed was donated by Georgia Crop Improvement Association, and of course the employers, University of Georgia and USDA ARS, for the PIs on this study. For any additional information, comments, or questions, you can contact me at tubbs@uga, and I certainly thank you for your attention today.